I am Poonam Chaudhary, Assistant Professor in Botany, Dronacharya Government College, Gurugram, under the aegis of Directorate of Higher Education, Haryana. The topic of my lecture is lysosomes, their structure and functions. So, let's start. The contents of this lecture are as follows. First, what are lysosomes? Then, structure of lysosomes. Next will be lysosomal enzymes. After that, we will study site of synthesis. That will be followed by types of lysosomes, then functions of lysosomes. Last but not the least, summary and frequently asked questions. Now, let's start. What are lysosomes? Lysosomes are an important cell organelle found within eukaryotic cells. Due to their peculiar function, they are also known as the suicide bags of the cell, or we can say, or we can define, lysosomes are an important spherical cell organelle filled with hydrolytic enzymes that have the capability to break down many types of biomolecules. The term lysosomes was coined by Christian de Duve, a Belgian biologist who discovered it and ultimately got a Nobel Prize in medicine in the year 1974. The word lysosome is made up of two words, lysis meaning breakdown and soma meaning body. In other words, lysosomes are membranous organelles whose specific function is to break down cellular waste and debris by engulfing it with hydrolytic enzymes. Now, structure of lysosome. Lysosomes are membrane-bound organelles and they show loads of diversity in shapes and sizes. The number is also variable in different types of cells. Lysosomes are usually placed near the nucleus. As we know, lysosomes are membrane-bound organelles and the area within the membrane is called the lumen, which contains the hydrolytic enzymes and the other cellular debris. The pH level of the lumen lies between 4.5 and 5.0, which makes it quite acidic. Lysosomes appear as cytoplasmic dense bodies that may be ovoid, spherical, globular, or granular. As the sizes of lysosomes vary, with the largest ones measuring in more at than 1.2 micrometer, but they typically range from 0.1 micrometer to 0.6 micrometer and surrounded by a lipid bilayer along with transport proteins. This membrane is unique in composition that prevents its components from being released into the cytoplasm. The lipids that make up the bilayer are phospholipids. The membrane contains highly glycosylated lysosomal associated membrane proteins and lysosomal integral membrane proteins. Lysosomal associated membrane proteins and lysosomal integral membrane proteins form a coat on the inner surface of the membrane. They protect the membrane from attack by the numerous hydrolytic enzymes retained inside. The lysosomal membrane has a hydrogen pump which is responsible for maintaining pH conditions of the enzyme. The acidic medium maintained by the proton pump that pumps hydrogen ions inside the lumen and ensures the functionality of the lysosomal enzymes. Enzymes in lysosomes. For degradation of extra and intracellular material, lysosomes filled with enzymes called hydrolases. Lysosomes contain about 50 enzymes which are classified into different groups. Proteases, which digest proteins, lipases, which digest lipids, glycosidases, which digest carbohydrates, nucleases, which digest nucleic acids, phosphatases, which act on organic compounds and release phosphate. Sulfatases also act on organic compounds and release sulfates. So, we can say collectively the group of enzymes is called hydrolases, which cause cleavage of substrates by the addition of water molecules. Most of these lysosomal enzymes function in the acidic medium. Hence, they are also called acid hydrolases because digestive enzymes usually function in acidic medium or pH of 4 to 5. These acidic conditions are maintained inside the lysosomes by pumping of hydrogen ions or protons into them. Now we will discuss site of synthesis of lysosomes. Lysosomes are manufactured and buried into the cytoplasm by the Golgi apparatus with enzymes. The enzymes that are within the lysosomes are synthesized into rough endoplasmic reticulum. Once synthesized, the enzymes are then delivered to Golgi apparatus via transport vesicles. Lysosomes pass through various stages in the same cell. This phenomenon is called polymorphism or existence of more than one morphological form. Now, types of lysosomes. Depending upon their morphology and functions, There are four types of lysosomes. The types of lysosomes are primary lysosomes, secondary lysosomes, residual bodies or tertiary lysosomes, and last one is autophagic vacuoles. First, primary lysosomes. Primary lysosomes are formed from Golgi apparatus appearing as small vesicles. Primary lysosomes, simply called as storage granules storing inactive hydrolases. These are not involved in digestive process. Primary lysosomes do not release any of their content. Next is secondary lysosomes. Secondary lysosomes are formed when primary lysosome fuse with phagosomes. The fusion also causes the previously inactive enzymes to be activated and capable of digesting biomolecules. Thus, they are involved in the process of digestion. Hence, serve as digestive vacuoles. Compared to primary lysosomes, secondary are larger in size and capable of releasing their enzymes outside the cells where they degrade foreign material. Third one is residual bodies, or we can say residual lysosomes or tertiary lysosomes. They are those lysosomes in which only indigestible food materials have been left. The residual bodies or lysosomes pass outwardly and fuse with plasma membrane to throw out the debris into external environment by exocytosis. Sometimes residual bodies remain inside the cells due to failure of exocytosis and absence of some hydrolytic enzymes. This leads to storage diseases like aging. Last one is autophagic vacuoles, or we can say autophagosomes and autolysosomes. They are produced by the fusion of a number of primary lysosomes around degenerate intracellular organelles. Autophagic vacuoles are used in removing internal obstructions and also provide nourishment during starvation. Now, the functions of lysosomes. The lysosome is essentially a bag containing number of digestive enzymes. The enzymes may be released within the cell itself or outside the cell. As we know, lysosomes perform several functions like as heterophagy, autophagy, extracellular digestion, autolysis, genetic changes. Functions of lysosomes one by one. The first function is heterophagy. Heterophagy is the lysosomal digestion of extracellular materials by the process of endocytosis. In endocytosis, invagination of the plasma membrane of the cell results in the creation of an endocytic vesicle that engulfs different types of extracellular materials. Heterophagy is of two types. First, phagocytosis. Second one, pinocytosis. Phagocytosis means cellular eating. It is an intake of solid particles with the formation of vesicles called phagosomes. Second one, pinocytosis. Pinocytosis means cellular drinking. It is the intake of liquid particles with the formation of vesicles called pinosomes. Lysosomes serve as intracellular digestive system, hence called as digestive bags. Lysosomes also help to defend against pathogen entry via endocytosis by reaching pathogens before they reach cytoplasm. Next function is autophagy. Autophagy is also known as self-eating or autodigestion. It is a normal physiological process that deals with the destruction of unnecessary or dysfunctional components of cells. It is essential for maintaining homeostasis, for normal functioning by protein degradation, turnover of destroyed cell organelles for new cell formation. Autophagy has great significance as during starvation, the lysosomes digest stored food contents such as fats and glycogen of the cytoplasm and supply the necessary amount of energy to the cell. They also remove the worn out and poorly working cellular organelles by digesting them to make way for their new replacement. In this way, they remove the cell debris and help in disposal of cell debris. Therefore, lysosomes are also called as scavengers and cellular housekeepers. Next one is extracellular digestion. Lysosomes secrete hydrolysis outside by exocytosis, resulting in degradation of extracellular materials. Example, saprophytic fungi. Next one is autolysis. Autolysis is the destruction of cells or tissues by their own enzymes that are released by lysosomes. In layman's language, we can say that autolysis is self-digestion. Autolysis means the disintegration of the cell by the destruction of its cell membrane. Sometimes the digestive enzymes may end up damaging the lysosomes themselves, and this can cause the cell to die. Therefore, lysosomes are also known as suicide bags of the cell. Last one is genetic changes. Due to the presence of nucleases, lysosome has an ability to harm genetic material through the release of nucleases. It may result in mutations, chromosomal breakages, and other abnormalities. Now, we will discuss keynotes. Keynotes of lysosomes are cell organelles found in eukaryotic cells. Lysosomes are membrane-bound spherical sacs filled with hydrolytic enzymes. The enzymes present in lysosomes can break down many types of biomolecules such as proteins and fats. Lysosomes are known as suicidal bugs as they tend to destroy the cell membrane with their digestive enzymes, causing lysis of the cell, that is called as autolysis. Lysosomal enzymes are synthesized in rough endoplasmic reticulum where they are brought in the Golgi apparatus through tiny vesicles. Lysosomes protect the cells from infection as they destroy any foreign material which enters the cell, such as bacteria and viruses. Now, in last, frequently asked questions. First, short answer questions. Question number 1. Define lysosomes. Question number 2. Why are lysosomes known as suicidal bags? Question number 3. Where are the enzymes needed by lysosomes made? Next question. Define autolysis. Next one. What are functions of lysosomes? The role of enzymes inside a lysosomes can be asked. Last one. Write a short note on lysosomes. Now, multiple choice questions. Question number 1. Which of the following organelle is called as the perinuclear dense bodies? Options are lysosome, nucleolus, peroxisome, all of these. Question number 2. The pH of lysosome is neutral, acidic, basic, depends on the cell type. Question number 3. The acidic pH of lysosome is due to options are deposition of waste materials, presence of hydrolytic enzymes which has an acidic optimum pH, presence of anabolic enzymes which has an acidic optimum pH, or all of these. Question number 4. Which of the following organelle is called as the suicidal bags of the cells? Options are lysosomes, lysosomes and Golgi, lysosomes and ER, lysosomes and mitochondria. Question number 5. Which of the following is the function of lysosomes? Options are autophagy, autolysis, digestion, all of these. Last question is, lysosomes are involved in extracellular digestion, intracellular digestion, both A and B, and none of these.